Right. So, again, hello from us. We'll present our SiDock at home project. So, drug discovery via distributed computing. We'll just start with why. Why did we actually go into this? So, of course, coronaviruses, everyday companions, very notorious because can skip from other hosts of humans. And of course, they are named after their crown spikes. So that's why corona. There are 4 major subgroups. And of course, they were discovered in mid-60s. And of course, they namely cause common colds, but this time something completely different happened. So, previous occurrences of related pathogens can be traced to 2003. And then coronaviruses were known to cause SARS and MERS respiratory syndromes. And of course, now in 2019 to 2020, a new pathogen was reported, initially 2019 nCoV, or now named SARS coronavirus 2. N was named, of course, for novel. And the virus spread from Wuhan, China. And of course, in the early 2020, the virus spread and caused a global pandemic situation. The situation is now quite problematic because it causes variable percentage of complications and of course also various percentage of death causes. Now, why and when did we decided to do SiDock at home? So of course we are a drug discovery team and work in this field in our main institutions. And of course we said global problem needs a global solution. And of course in December already we started to follow the media and media reports. And in January, of course, we had over beer discussions. In February 2020, we set up a web presence. And of course, we also developed COVID SI community and even our own client that was meant to help us compute everything. And of course, work was February, March already underway. And in April, first results started to drop in. Again, in May 2020, we got full support from University of Maribor and University of Primorska. In June, July 2020, we got a wonderful team expansion where Natalia and Maxime joined the project. We then got a bit of commercial support. Microsoft got contact with us, but they enforced really the sales of their tools. So of course we were very grateful for their support, but were really stumped what to do because we had a lot of data to process and great challenges for the future. And of course then turned to BOINC. In October 2020, the first initial BOINC setup was done. And of course, up to now, the project organically grows. So what are we actually doing? So a little bit of medicinal chemistry and in silico chemistry for all of you. So we are actually doing virtual screening. I couldn't resist putting this funnel on this slide because we are here in Slovenia. It's a wine country and we all dream of these funnels. I don't know why, but actually the process is like this. So in essence, we are trying to screen and to pour a lot of compounds into some sort of filter funnel and then identify compounds that could have good propensity to binding and could have some potential for their optimization. So of course we are looking for structures that are most likely to bind to a drug target, and that target is typically a large protein or receptor or an enzyme and similar. So what is actually going on? So the specific action of an enzyme with a single substrate can be approximated with a lock and key model that was initially postulated by Emil Fischer. So actually you see here a ligand, a small molecule, maybe a drug, and of course a large molecule, a protein target, and that ligand binds into the protein and of course forms protein ligand complex. And we are looking for such small ligands that are able to bind in this large target via some specific interactions that can form between the protein and the small molecule. So what is actually an input for this experiment? So I just referenced an article. So we are actually searching for a chemical space, and the chemical space consists of a large body of organic molecules. Some say that the space is of the size 10 to the power of 60. On the top right corner, I have just a small graph just showing the drugs, known drugs, in black color, and the green triangles are natural products that are known. So you can see just how small fraction of those drugs are in the context of just natural products. If we plotted the whole chemical space of all organic compounds, it would take all this green. And even in this, we are searching for really a small subset of compounds that can bind to our target. So here are some with green circles, and red and blue circles are emphasized some key interactions such as H bonds and of course some lipophilic features. But in general, we can say that the small molecule must be complementary in shape and charge to its target. So what we are doing here, it's a selection of proteins that can be found in this new pathogen, new coronavirus. So this is its genetic sequence and all the proteins that are encoded by that genetic sequence. And some of these proteins are actually useful as a potential therapeutic target. And we, of course, started to study the genome and selected a couple of prospective targets to work on. Okay, this is just a scheme of the virus known to everybody, present in all media, and just a couple of proteins. So M for membrane, E for envelope, S for spike protein, and N for nucleocapsid. So the most known is, of course, S protein, known to everyone, spike, because it is responsible for binding to the host receptor. So what is actually virus doing? So from the top left hand of the screen, the virus is binding to the receptor, typically AC2. It enters the cell. So what is colored blue is actually our cell, so host cell, human cell, for example. So virus enters our cell into the endosome, it uncoats, then it is translated to polyproteins. These are large proteins, and these are then proteolyzed to smaller proteins that are actually functional, viral and structural proteins. Of course, then via transcription and translation, new proteins are formed, and via replication, also viral RNA is replicated and prepared. And after formation in Golgi apparatus, the virus is reassembled or assembled, and of course, in the end, released out of the cell. So in these orange boxes, we highlighted the targets that we are working on. So we're working on entry, S protein, AC2, on proteolysis, on transcription, replication, and of course viral assembly. And we will try to help the scientific community in these fields. So, of course, the first targets we chose are viral proteases. So virus uses its proteases to process initial translated polyproteins into functional protein molecules. And of course, we selected currently the mostly researched 3CL-Pro or viral main protease. It's a cysteine protease, and we are really working hardly on it, and of course have also first results. What are our goals? Or design small molecule covalent and non-covalent inhibitors of this protease. So on the left side, this is catalytic diet of the enzyme, what is actually the enzyme doing, and on the right you can see two molecules that are known to be inhibitors. So these are small organic molecules. These P1, P2, P3 are pockets in the protein target, and cysteine 145 at the bottom is actually catalytic residue of the protein. So how does this look like in the actual protein? The protein looks like this, and in this box you can see the active site and one of the small molecules in the active site. And when we dock various compounds, we try to identify which one of those could fit into this active site. So it looks somehow like this. So this is just a swarm. It looks like a small haystack of small molecules that can bind in one way or another to the active site. And we are trying to find the ones that are really favorable. So it looks like this. And these are some examples of compounds that we identified that are of potential. And of course, we are already in the phase of purchasing and in acquiring of actual physical samples, and laboratory tests are already underway for the first target. So for the second target I mentioned, we also work on other viral steps. So this is RNA-dependent RNA polymerase. Again, this is the protein. With red, it's depicted its active site, and with yellow, a small molecule that can actually bind to that active site. And on the right side are a couple of drugs. You will immediately recognize remdesivir. On the bottom is actually a prodrug that gets changed to the active form GS441524, and it looks like that, and is an inhibitor of course of RNA-dependent RNA polymerase, and actually can stop the binding of the nucleus and RNA. And of course, we are heavily working also on this target and performing virtual screening on this active site that is concretely depicted here. And how does the whole workflow look like? So, of course, we have to do compound preparation. These are billions of compounds. And then we go to HTVs. It means high throughput virtual screening. And this we do through BOINC, where you community helps us. And I must emphasize that we have a full stack from HTVs software tools to BOINC. So we also are in a unique position that we are developing the tool for molecular docking itself. So it is CMDOC or KiriMariDoc. I will explain a bit later. And after the results, we do post-processing. And of course, this is RNA-dependent RNA polymerase. You can see RNA chain here near the active site. And of course, we have to select the target, select the active site. We do additional MD experiments to really verify that the target is suitable. And of course, we do post-processing of the small molecule protein complexes. And after that, we do hit selection and bioevaluation. So for two viral proteases, 3CL-Pro and PL-Pro, we are in this stage. So compounds are underway, and wet lab is in working. And of course, this cycle is meant to be progressed and iterated. And of course, HTVs again done with the help of BOINC. And of course, we have two servers, Xeon-based servers with 36 and 56 threads. And of course, we had 500 active computers in the testing phase and more than 3,300 active computers in the main phase after 4 months. And I'm really happy. Just today, me and Chertomir installed our new server dedicated BOINC server that is so fresh that it's fuming right now, hopefully from the freshness, not from the burnout. So yeah, okay. So software for molecular docking, we used RxDoc and of course now CMDOC, and BOINC wrapper to adapt the software to the BOINC environment. And this is the evolution of our project. So of course, it's really nice here to see the end of testing phase where registered hosts really jumped. And of course, we then had BOINC BAM challenge. Of course, registered hosts jumped again to 6,000, and BOINC formula sprint, we again slowly we are accumulating users to now reach almost 12,000 registered hosts. So the project is really organically growing. Work progress in half a year. So we screened 1 billion compounds for FreeCL-Pro. This is the viral main protease. Then we screened 1 billion compounds against 2 PL-Pro viral proteases. These are 2 binding sites distinct on this viral protease. And then we screened, of course, 1 billion compounds roughly to the target RDRP, so RNA-dependent RNA polymerase. This is also completed, and now we are screening for the E-protein test targets. And of course, we are continuing our work. And for the first 2 targets, so FreeCL-Pro V3 and PL-Pro V1, as I said, wet lab is actually in process. So bioevaluation experiments are already completed. The systems are ready. So we are actually just waiting for the compounds, and those are connected to costs. So of course, compounds have to be purchased and so on. And that's why we need a bit more time, but actually we are proceeding quite well on this front. So current work moving to a new server. Actually, we moved to a new server thanks to Natasha. We are working on the checkpoints, and they are actually implemented thanks to Gasper. So with the great thanks to BOINC community, and we are working hard on GPU version of CMDOC. So CMDOC, it's actually a collection of optimizations, quality of life, and parallelism updates on a fast and versatile open source docking software. And we are really committed to this project. We invested our whole life into it, and we will really work hard to work on this CMDOC and to bring something new, not just to the community, but also to the medicinal chemistry and in silico computational chemistry in general. What can it do? So here is an example of protein with the mesh, blue mesh. You can see an active site defined for docking, and these are crystal structures and redocked structures for reference. So software can really perform well and produce meaningful results. So it can prepare targets. It can run from detailed dockings to HTVs, and of course it can design HTV protocols. It can do analysis of results. We have a full suite of SDF tools, courtesy of Gasper, and of course in active development, and we will be committed to support this software and of course support BOINC to the end. What next? So viral protein E is actually in process. We are working on it. Of course, S protein, we already are working on it and preparing the targets. So how to work on the viral entry, and of course transmembrane protease. Of course, we will work on that too. And on the left side, this is a beautiful protein. So in colors is S protein, and in magenta is AC2 receptor. So here is the team. Special thanks to Chertomir, Natalia, Maxim, and of course Gasper. And we especially thank Stefan Müller for bringing us all together. He has a special place in our hearts, and we hope we'll work with him more in the future. And the most important part of our team is all of you. So participants of SiDock at home. With your help, SiDock is becoming a general drug design project and aimed at the future problems. So we will try to work on pan-corona medicines. We will continue our work on Ebola medicines. We have a joint project on malaria drugs, and of course we'll try to work on oncology targets in the future. And with that, I would like to conclude. Thank you all for listening and especially for your attention. And again, sincere thanks to all of you from our hearts. Thank you.
Thank you very much. Thanks, Marko. Any questions? Please use the hand raise feature under reactions. In the meantime, actually, Marko, I have two questions for you. One is, yes. You mentioned in your workflow the docking and also molecular dynamics. Are you doing the molecular dynamics on BOINC also?
No, we are not doing molecular dynamics on BOINC. We are doing it on our own systems. We are mainly using it to produce better targets for HTVs on BOINC.
Okay. Yeah. And about CMDOC, is that open source? Could other BOINC projects use that? Or
Of course, you are welcome to go to its repo, download it, and please use it and report to us what can we do better.
Okay, you said it was optimized. Is it an optimized version of like Vina or Smina or something like that?
No, it's completely different software, and it's actually a optimized version in regards to previous versions. And we are working still further, and especially, of course, on GPU.
Okay, great. So last chance for questions, and oh, we have one from Stefan. So let me.
Yeah. Thank you for the praise. Question is, of course, when we have the AutoDoc team here now, among participants, they haven't asked currently, but when you compare with AutoDoc Vina in particular, so what are the main differences for the interpretation of the results that you get?
Yes, so they are, you could say, similar softwares in the sense they are both molecular docking softwares, but they are quite different. So okay, they use genetic algorithm for pose optimization, but they use completely distinct scoring functions, and they use a distinct input of data. So the input pipeline is completely different. Of course, they are similar, but on different systems they behave a bit differently. And when we started work on this subject, as a medicinal chemist, usually use many tools at the same time, and we tested it and really performed quite well, this software. And of course, we decided to use it. And now, as we know it more, it is quite performant, and we are very pleased with the results. So yeah. We are quite pleased how it goes.
Stefan, does that address your question?
Yeah, many things. I couldn't unmute myself. So yes, it does. And well, so there's no competition in the sense, right? There's just competition for users now who either crunch for fight AIDS at home, or what was it, like open academics, or they crunch now for your project. So yeah, let's see how this goes, and keep up the good spirit. I liked it. Nice presentation. Thank you.
Yeah, thank you very much. We are really a young project. We are really underdogs, you could say, but we are a dedicated team, and we will do our best to push this forward to indefinite.